So today we will start off with our first speaker, Joshua Dibble from the University of Edinburgh. Joshua, whenever you're ready.
Actually, I might just share my screen as normal. Okay, can you all see that? Could I get confirmation from someone?
Yes, we can see that. Thank you,
Awesome.
Joshua.
Thank you very much. Okay, so thank you for having me. I'll apologize in advance. I'm very jet lagged at the moment, so I hope I'm going to get through this whole presentation mostly coherently. So I've been working on the Decode project, which hopefully you've heard of. We published a preprint a few weeks ago now. It is to date the largest ME study based at the University of Edinburgh, but also in collaboration with various UK organisations, including charities and some governmental funding as well. I thought, if you've heard of the project, you probably expected Chris Ponting to be giving this presentation, and indeed he spoke earlier this week at the closed meeting. Unfortunately for you guys, he's on vacation, so you're stuck with me instead. So I thought I'd give you a brief introduction to the stats team that have done most of the analysis over the last few months. So I did my PhD with Chris at Edinburgh until 2022, when I moved to the US, and I was a research fellow working with Wenjong Zhao, who I can see is on this call, and I believe Dr. Sistrom, who I collaborated with, is presenting later as well. Early this year, I moved back to the UK, and Chris offered me a supposedly short-term position working on Decode. But as I'll explain throughout this presentation, while the project has now come to an end, there is still a lot of work we're going to be doing. So I'm going to carry on working with this data for a while. Yeah. Most of the work done in the slides I'm presenting today was either from myself, from our team lead, Thibaut Bertin, or from Esther. Most of Esther's work will actually be in the published version, but everything today is largely the result of our collaboration. There is a larger genetic delivery team, which is fully credited in the paper. If you haven't already, please do go and take a look at it. We put a lot of work into it. So, summary of what I'll go through. We have the results are now, or at least the initial results are now public. So I will talk a little bit through those. But really, I think I'll try and focus more on what they mean, because you can go read the p-values and the tables and the plots yourself. But I think an important step is actually going through the implications. But I'll start at the beginning and talk about why we've looked at genetics and how we got to this stage. And then I'll talk a bit about what's next for the DecodeME project. As I said, it's just officially come to a close. And then what's next in terms of future projects that could use these samples and this data. I don't know if I have Q&A time, but I'm happy to take questions via email or something if necessary. So why genetics?
Okay.
The way we do it, population. Oh, sorry. Okay, anyway. The way we do it, population level, it's unbiased and it's comprehensive. We don't go in with a prior hypothesis based on biology. There are many symptoms of ME, and you'll hear lots of talks today that focus on them in turn: the muscle and joint pain, the immune system effects, CNS, the autonomic symptoms like POTS, and then the endocrine dysfunction that many people report. We could, in theory, have used our £3.2 million grant to work on one of those. Instead, we've decided to look for, I think, as our media team put it, the treasure map. We want a better and more systematic way to focus on these, to find which of these we should be focusing on. There's also evidence to suggest that selecting genetically supported targets could actually improve the success rate of clinical treatments. So although everything I'm showing you today will be very abstract from a treatment and diagnosis perspective, it will hopefully accelerate the later processes. So we know that multiple members of the same family can be diagnosed with ME, and I think both anecdotally, I know most of this audience will have some experience of the disease. It seems to run in families to some extent, but it doesn't follow a predictable Mendelian pattern, by which I mean it's not like cystic fibrosis or Huntington's disease, where if one of your parents has the genetic variant, then you can predict with probability whether someone has it. If both of your parents have the recessive copy, then you end up with the disease. So we don't think that's going to be the case with ME. Certainly, we've never found any evidence of that. So we think there'll be many individual regions that will slightly alter the risk. Decode, we found that the amount of risk that's heritable seemed to be around the 10% mark, based on markers from the microarrays. This is slightly more than we would expect for a trait. Please don't quote me on this because I don't have a reference for it, but I think it's about 8% for all traits. So we are slightly above what we would expect for most genetic traits. Oh, excuse me. So the way we do this is something called a genome-wide association study, or GWAS. We are taking DNA and we look at the differences at individual points along the genome. So we might say at this position, most people have a C, but in some people it's a T. And what we're trying to figure out is whether this change is more or less common among people with ME. We have large numbers. So as I said, largest study on ME, we have, after all of our various filtering steps, which I'll go through in a second, we have about 15,500 cases and about 250,000 population controls taken from the UK Biobank. One technical point I'm going to mention on this slide, because it becomes quite important later, is this is not whole genome sequencing. So you've probably heard of whole genome sequencing, which is literally measuring almost literally measuring every base on the genome. That is very expensive, and we didn't have the funding to do that. We've instead used an older technology called arrays, so where we sample across the genome, and then we use something called an imputation algorithm to make educated guesses, almost, to try to predict the missing bases. This, as I said, I'll come back to this, but imputation, when I talk about it, is this mathematical process of predicting the missing bases. So here's a good example of what a GWAS, I guess almost the best case scenario for a GWAS. So this was from Lyme disease. So they had 25,000 Lyme disease cases and almost 600,000 controls. There are 3 very clear hits on this plot. So you can see that gray line. I don't know if you can see my cursor, but there is a gray horizontal line, which is at 5 times 10 to the minus 8. We call this the genome-wide significant threshold. For autoimmune and immune diseases, it's very common and almost expected that there will be a hit on a region called the HLA on chromosome 6, human leukocyte antigen. There is one for Lyme disease. This study also found a hit for TLR1, which had previously been associated with Lyme disease. But the most interesting is this enormous signal on chromosome 11. So this is the paper. And it's the central point of their paper is that this hit they found, SCGB1D2, inhibits the growth of the bacteria that causes Lyme disease. So this is kind of the ideal that we would find from a GWAS. This gene or this protein is expressed in, I think it's in skin or something similar. If you have this variant, then it prevents the Lyme bacterium from growing, and it decreases your risk of Lyme disease. So it's not that the variant is directly causing or preventing the disease, it's that there is a clear downstream mechanism we can follow. And I think this paper actually did some of the wet lab experiments to prove this. So this would be best case scenario. But to get to that point, we actually needed a cohort of people with ME, and we had to build our own because there isn't one of this scale. So we asked for people to volunteer. It was a 23andMe style, if you've ever used or heard of that program. Spit a test tube, take a saliva sample. We used a post-based design. So they would do this at home and then mail them back to us. Participants had to live in the UK for practical reasons with this kind of posting design or mailing design. They had to have an ME diagnosis from a physician. So they were self-reporting, but they should have been reporting a physician diagnosis, not a self-diagnosis. And they had to be over 16 because we were interested in the adult form. So our key objectives were to identify the genetic differences in people with ME and hopefully to start explaining the biological mechanisms. We wanted to build a clearly defined cohort of people with ME for future research, so for our internal projects, but also we want to be able to disseminate this data to other academics and to industry partners. Everyone in the consent form had the option to have the data made available for other researchers. And I have a slide right at the end where I'll talk a little bit about the access process. Please do get in touch if you think you can use our data. This project was co-produced with the ME community. I believe this got some kind of gold standard award for PPI. We had a full PPI steering group, particularly for the early stages of the project when the questionnaires were being designed and all the samples were being collected. They were a huge influence on the project and yeah, were absolutely vital. We managed to recruit in the end over 20,000 people across the UK. And I've got a breakdown roughly of the stages here. So we had almost 40,000 people sign up. Just over 20,000 completed the question. Sorry, 26,000 completed the questionnaire, like the initial questionnaire. From that, 21,000 or so met the eligibility criteria to proceed with the genetic testing and were sent the collection kits. About 18,000 of those returned their kit. From those, we managed to extract DNA from 16 or 17,000, and those are the ones that then subsequently passed genotyping and QC. But sorry, there should be an intermediate step there, I guess. So that was all of the ones that came through the extraction, genotyping, and QC processes. And then we filtered to cases of European ancestry. I will talk about this in a second. It's vital for GWAS that your cases and controls are matched as closely as possible. And the UK Biobank skews heavily British just because it was done in the UK. So this was not done based upon their self-reported like background or anything. We did this purely based on genetic similarities. You can see here there are some plots showing comorbidities, and the age peak, our age was about, the peak seemed to be in the sort of 50 to 60 range for us. Here's a very quick walkthrough of the QC steps we did, just to give you an idea of how complicated this was and why it's taken so long. The project's been running for about 3 years now. So first off, we had the sample QC, so each individual sample had to pass QC procedures. We then did an analysis QC using Axiom software. This is the same, approximately the same procedure as used by the UK Biobank. We had to match every step in our cases that had been done for the controls. So this was done using some software to find like outliers in various ways. We also corrected for plate and batch effects. So you have to account for the fact that everything has to be broken down into smaller groups for processing. And I mentioned that imputation step where we're mathematically predicting the missing bases that we basically didn't have the money to do whole genome sequencing on, and we filtered anything that wasn't sort of 90% confident. It's called the info score. And there was another technical step called the Hardy-Weinberg equilibrium, and we filtered based on that as well. So there have been months and months of very careful processing to make sure these samples were the best possible to put into the downstream analysis. So I talked or mentioned briefly that we were matching by ancestry, and I've kind of wanted to slide on this just to emphasize the point on what we were doing. So sorry, there is a typo here. That should be controls are shown in gray and cases are in green. So the key points are all of the gray dots came from the UK Biobank and all the green ones from DecodeME, and we wanted these shapes to match as closely as possible. Any mismatch could lead to false positives when we actually do the analysis, and we don't want to find results that are being driven by ancestral differences rather than by the difference between having ME and not having ME. So broadly, I won't talk too much about principal components analysis, but broadly, these PCs reflect sort of the greatest differences in each sample. So in UK Biobank, principal components 1 and principal components 2 broadly match the self-report for white British and white Irish. In a genetic study in the UK, those are the two main population groups. Later ones sometimes reflect technical steps and similar, but we were broadly trying to make sure that the ancestry is matched as closely as possible. So this is the key slide from the entire presentation. We discovered 8 signals. These are spread across the genome. We have a hit on chromosome 1, which we are annotating as RABGAP1L. We have 2 from chromosome 6. One is BTN2A2. The second is FBXL4. On chromosomes 12 and 13, we have SUDS3 and OLMF4. 15 is CCPG1. 17 is CA10. And 20 is ARFGF2 or CSE1L. I've gone through them one at a time because you probably noticed that one of them has 2 annotations. The art of annotating these is not perfect. So we have found individual variants. So I showed you the DNA sequence. So an individual change in the DNA. And the sort of difficult downstream step is then to figure out what that is actually doing. Usually, the change will be affecting the closest gene, but not always. So some of the stuff I'll talk about in a moment was about finding the most appropriate label for this plot for each of these variants. And the reason chromosome 20 has 2 is because we're actually unsure which of these 2 genes is most likely to be affected. So everything above this dashed line indicates a single base change in DNA that was significant statistically in our study. They're called Manhattan plots because you can say, well, vaguely looks like skyscrapers. So these 8 signals were found spread across 4 GWAS. This is slightly complicated, but we had a primary study called GWAS1. This is all explained in detail in the paper, but I'll try and cover it quickly. So we have a main study called GWAS1, which found 6 of those associations, the ones on chromosome 1, 6, 15, 17, and 20. We then defined a second cohort called GWAS2, which will form part of our replication study. This is still ongoing, but this study also found 2 additional hits on chromosomes 12 and 13. And if I tap back quickly, you can see the skyscrapers for those genes, for SUDS3 and OLFEM4, are just below our line. So we're principal scientists. We don't call these significant, but we have found evidence in other cohorts. So our GWAS2 cohort and a GWAS cohort defined by those who answered that they had an infection before ME. We call this the infection at onset cohort. These signals are actually above that dashed line in those studies. So we're annotating them as though they were hits. So we're calling it 8 signals. About 70% of people, I think I quickly added these up just before the presentation, something like 60 or 70% of people report an infection either when their ME symptoms started or just before. It's typically glandular fever, so mono, or other, which probably includes flu and similar as well. What do these genes do? Broadly, we think they fall into 2 categories. We think they affect the nervous system. So the one on chromosome 17 is actually, we think, the same association as one that's previously been found in chronic pain. So this may not be ME specific, we're not sure, but we were encouraged to find that we'd replicated a relevant previous phenotype. And indeed, one of our other analyses, something called MAGMA. So I didn't do this. This was Thibaut's work. So I'm not going to talk about it too much. But just to say, to sort of support our idea that the nervous system is involved in some way, this MAGMA gene tissue analysis showed significant enrichment in all 13 brain tissues for ME-related genes. So we're sort of building the weight of evidence that we think the nervous system is involved in some way. The second main set we think are involved in the immune system. And these likely reflect the infection before onset, because many of the people in our study did report an infection before their ME symptoms started. The 2 clear ones were one of the 2 chromosome 6 hits and the one on chromosome 13. But there is an additional type of immune system. So these ones we think are largely involved with the innate immune system. So the HLA region that I mentioned briefly when talking about Lyme disease, and is often involved in immune-related disorders, we found one association, but there is a caveat, because this was not done in the same way as all the rest of our studies. So we're treating this with a large grain of salt. So for the HLA analysis, for the main GWAS, we imputed all of our cases and controls together to avoid technical artifacts. For the HLA, it's a much more complicated region, and we attempted to match the UK Biobank analysis, but the UK Biobank paper came out in 2016, so the software is quite old, and it's quite difficult to run at scale. We literally ran this on cases only, so 16,000 or so people on 4 desktop computers in the office. So trying to do this for our controls as well was just computationally intractable. It would have taken months. So we took the UK Biobank's pre-provided imputations for the controls. We used our own ones for the cases, and we did the analysis, and we found 1 hit. We're skeptical somewhat, because this, so it's HLA DQA10501. This is usually found alongside 2 other associations, and we didn't detect those. So we're unsure whether this reflects something ME relevant, or whether this is just a technical artifact of the way we've done the analysis. So we've obviously published the preprint, but we haven't submitted to a journal yet. One of the priorities before we submit to the journal is we're going to redo this completely, ignoring the UK Biobank method, and we're going to re-impute all of their controls as well with a modern algorithm on a cluster, so on a high-performance computer. So still immune-related, possibly relevant, but we have more work to do on this one. I mentioned the issues with labeling the sort of peaks on the Manhattan plot, and this is somewhat technical, so I'm going to skim very lightly over this. Essentially, the way we do this is we have a separate data set called GTEC, which gives us information on gene activity, and we have our ME-CFS genetics. And what we're trying to do is determine whether the variant we found in ME, so this sort of green label here, is the same as the nearby ones that regulate gene activity. The package we're using is, or the technique we're using is something called colocalization. It's a Bayesian approach. And what we're aiming for is this example on the right here, where we find that the variant we found is exactly the same, or overlaps with the region that regulates gene activity, and then we can, with reasonable confidence, say that the variant we found is related to that gene, and we then label the plot. I'm quite proud of this plot just because it looks kind of cool, but this packs a lot of information into quite a small area. So this is the output from that process I just described. So all of the gene names are on the left-hand side, and all the tissues are on the horizontal axis. So big dots indicate high probability that the variant we found in ME is the same as the one regulating that gene in a given tissue. So to give you an example, this blue dot here for RABGAP1L is is likely the same, is expressed in subcutaneous adipose, so in fat tissue. And we think there is a high probability that the variant we found in ME is related to the gene expression, to that gene's expression in that tissue. The way we annotated the plot was to look at how many dots, basically, so how many tissues is that gene expressed in, and we used that as our metric for which was the most likely if the information was available. So big dot high probability, little dot is some probability, no dot at all is probability of 0. So you can see for the chromosome 12 association, SUDS3 was chosen because there are a lot of dots all the way along, so it's expressed in a lot of tissues, and it's highly probable that it's the same variant as the one we found. Green indicates increasing risk with increasing gene expression. So if someone has the ME risk gene, then we expect that to be correlating with increased gene expression. Blue indicates the converse, so decreased gene expression. So this is the same technique, and it might be a bit easier for you to visualize what's going on with this one. So this was looking at other traits. In particular, we looked at chronic pain and depression, and we found no colocalization. So you can see the green here, again, is our DecodeME data. Gray is the other data resource. So this would be another GWAS that had looked to that trait, and we're looking to see whether there's an overlap with the signals they found. So there was a signal near ours to do with depression on chromosome 1, for example, but you can see the signal, so this is the panel B, you can see the signal is distinct from the green DecodeME signal. And you can see this visually, I think, but we're scientists, we don't do visual. The colocalization technique I mentioned demonstrated this as well, at least as of speaking today. For panels E and F, I can zoom in and show them in a bit more detail. This one did colocalize. So I mentioned that chromosome 17 here was associated with chronic pain. You can see here in panel E that the signal, the gray signal indicating chronic pain, is directly underneath the green signal indicating ME from DECODE. And when we zoom in on that region for panel F, you can see the structure looks very similar, in particular this dark blob on the right. Again, hopefully you can see my cursor. There is a green blob here and a gray one directly underneath it. And if we look at just that signal, we think it colocalizes. So we're confident that the signal we have found here is associated with multi-site chronic pain based on our research and the published literature. So to summarize the results so far, we have 8 genomic regions associated across those 4 individual GWAS. Evidence suggests so far that they are relevant to the nervous and immune systems. Further work ongoing, particularly with the immune system. We found no evidence for any sex bias, interestingly, among all these discovered associations, despite the sort of marked and noted sex bias in ME prevalence. About half of those 8 signals had modest replication in data from 2 other biobanks. So we worked with some collaborators that did some meta-analyses. They were significant at p less than 0.05, so the sort of standard threshold, but not the corrected one, I believe. So there is some evidence of replication, but really to see this properly, we would need several $1 million again to really do a sort of similar study in another country. Interpretation. What do these results mean for people with ME? So we've had a lot of questions, and Chris Ponting kindly provided me with a summary of the common ones and some sort of suggested answers. So what do they mean for people with ME and their families? Not much at the moment, because although the relative risk is, I think, 2, that reflects a doubling of risk, the risk is still small. It goes from about 1% to about 2%. So as I said, this isn't like a Mendelian disease where it's guaranteed with certain probability that you will pass it on to children. Does having these differences mean that you definitely have ME? No. Think of it as more of a tipping point. So there is some kind of environmental factor, as we said, it's usually like a virus or some kind of infection, and there is some point at which you tip over into having ME. These genetic differences, some of them at least, move that threshold, we think. So it may increase or decrease it. It may protect you from ME, some of them, or it may increase your risk of ME. And so the converse of that statement is also true. If you don't have them, it doesn't mean you can't get ME. That tipping point, we think, will probably still exist for everyone. It just may be that the threshold is much higher for some people. Does it mean ME is a life sentence and you won't recover? No. As I said, this is about risk, and the risks are quite small. We also haven't done a GWAS looking at recovery rates at all. What does this mean for treatment and testing? Nothing yet. This is very early days. Chris Ponting has been saying it's the end of the beginning. We have some very firm evidence now, but we can't assemble these into a panel for clinical testing. What does this mean for other conditions? To be tested. So we're looking, I think, at fibromyalgia in particular, but work is still ongoing for that. We can only speak about ME at the minute, not necessarily about long COVID and other diseases. If you're a participant, I'm not sure how many people in this audience will be, because I think it's largely US based. But if you were a participant, does it mean you have these you definitely have these differences? No. You may have some, you may have none, you may have all of them. This study is at population scale, and it doesn't have implications for individuals. What's next? So better gene prioritization. So we want to do more fine mapping. I sort of mentioned when I showed you those plots, we think that sort of blob, the green and gray one, are overlapping, but there's more work that can be done with better tools. Another priority is chromosome X. This is much more complicated than the autosomes, and we're still finishing that now. The HLA testing I talked about, the joint imputation, is another priority. And finishing the replication, we call them, so I talked about GWAS 2, we have the 3 and 4, which will be replication sets using biobank cases and controls. I've said this was only on people of European ancestry. So one of our other analyses, which is, I believe, running at the moment, is something called knockoff GWAS, which will be able to test all cases without filtering to ancestry match. The algorithm, the sort of technique works better with ancestral differences. And we are using 2 additional algorithms for sort of different perspectives. So they're called Targene and another one called GATE. They are, I believe, running or have just finished. Can't really talk about those at the moment. Final step, we'll submit to a journal and go through any subsequent revisions and resubmit if necessary. This is going to be months, unfortunately. We still have quite a lot that we want to do. But yeah, that's kind of our roadmap for the next few months. With samples, we have focused mostly on this area indicating common diseases and common variants with small effect size. So all the relative risks here are quite small. What we want to start looking at are rarer variants, which may have a larger effect size. So this is not Mendelian. We don't believe this is Mendelian, so we don't do sort of family-based approaches, but we can look at rarer variants with greater effect sizes. Unfortunately, this is quite expensive, so we are fundraising for Sequence ME, as we're calling it at the moment. So last couple of slides, just to say that we are as open access as we can be with genetics data, which I'm sure you're aware is reasonably tightly controlled. There is an applications process. If you're interested in working with our data, please do get in touch. Yeah, there's a full process. There's a committee that will review the application and will then hopefully be able to provide you with access to the data where participants have consented for that. Okay, finally, I need to thank at least some of the people involved. This has been a multiple-year project, and I joined fairly late. So there are some people on this list whose contracts started and finished before mine. This is also not a complete list because there are some collaborators that started more recently, particularly Hannah Lilla's team, who did some of the replication stuff with us, that are not on this list. We need to update this really with everyone, but thank you to everyone involved, including all the participants. And finally, yeah, as I said at the start, I am happy to take questions, but I don't know whether there's been time allotted for Q&A. Chris Ponting is away on vacation for about 3 weeks, but has said you can reach out to him. We're not monitoring the official DecodeME address anymore, unfortunately, just because the project's come to an end. But you can either contact him directly, or I'm happy, particularly if you have questions about this presentation, for you to write to me. I'll leave my email on the screen. Thank you very much.
Thank you, Joshua. That was great. Really appreciate it.